Hello everyone, and welcome back to Biopedia and our second episode covering cell polarity. I wouldn't usually make a link this explicit, but given how big the topic of cell polarity is, I thought it would be best to emphasize this connection in order to avoid getting lost. Today's topic is localized mRNA, with a bit of an introduction of what mRNA actually is along the way. Messenger RNA, or mRNA for short, is the intermediate molecule between the information encoded by DNA and the realization of those genetic instructions by proteins. When a protein needs to be created, the DNA unzips locally and RNA monomers bind through complementary base pairing to the DNA to create an RNA strand. This strand is then edited to remove any non-coding regions and stabilized. This is messenger RNA, which goes on to be translated into a protein. I won't go into the details of this now, as I want to stay on topic, but suffice to say we will cover mRNA and these intermediate steps in more detail in the future. Because an mRNA sequence codes for a specific protein, localizing mRNA to any one region of the cell ultimately means that a particular protein is expressed in one part of the cell, but not in another. This leads to a form of polarity, whereby one side of a cell or organism is consequently distinct in some way, shape, or form from the other. One example is in the common fruit fly, Drosophila melanogaster. There are principally two genes that have localized mRNA as the embryo develops, known as the bicoid and OSCAR genes. Through a mechanism that is likely too complicated to delve into for the moment, these two genes end up determining which end of the fruit fly will become which. The presence of the bicoid gene ends up determining the anterior, or front, end of the fly, while the localized presence of the OSCAR gene will determine which side becomes the posterior end. There are many different types of cells in our bodies which make use of localized mRNA in this fashion, although needless to say, this example of the fruit fly has skipped over some nuance. Another example occurs in the fibroblasts of human or chicks. A type of mRNA called beta-actin mRNA localizes to one side of the cell towards the edges. This is useful because the actin subsequently produced will go on to be used within a structure that will be covered in a future episode, the lamellipodium, which makes up part of the actin cytoskeleton. This structure helps cells to migrate by generating pressure against the plasma membrane. As a cell needs to move in one specific direction rather than trying to move to all locations at once when it is moving, this structure needs to be polarized. Hence, the localization of mRNA allows cells to direct themselves in one particular direction, an example which demonstrates the usefulness of localized mRNA in polarity more generally. As always, thank you for listening and tuning in with the podcast. We might well come back to some of the specific examples of localized mRNA in the future, as well as some other topics mentioned today, but for now this has simply been a primer episode to get us started on the topic, so we won't go into any further detail as such. For any queries, topic suggestions, or otherwise, the podcast can be contacted at biopediapodcast@gmail.com. Have a great week, and until next time.